Thank you, and thank you very much for inviting me and Matt and others, explaining to me what home visiting is about, research and home visiting. And I said initially, you know, why do you want me? And he said, well, we're trying to learn about other fields and what they're doing in terms of precision medicine. And cancer, of course, is all about precision medicine these days, as we're learning more and more about tumors. And you talk about heterogeneity, the heterogeneity of tumors, even within a particular tumor type, say breast cancer, is hugely heterogeneous. And it's not one size fits all. And how do we figure out what benefits individual patients? It means that clinical trials can't be as big as they used to be because there aren't enough patients to fill up any particular bin as we slice and dice the disease ever more finely. So I'll tell you about one thing that's going on in cancer, and all of what I'm telling you is new. I mean, it's as new to cancer people as it is to you. My disclosure, and you heard about this. This is the status quo of clinical trials in medical research generally. There's a clinical trial. At the end of the day, you calculate a p-value, and it applies to the population in the trial and the treatment that was used in the trial. Usually there's one question, you know, is A better than B? There's no modeling across endpoints. So in cancer, we look at tumor response, we look at progressive disease, we look at overall survival, and if you read an article in the Journal of Clinical Oncology, it'll tell you about the therapeutic effects in those particular endpoints, but it doesn't tell you how they fit together. And that's critically important, and it's important to you as you're looking at longitudinally within the patient and modeling that process. You can see, and I'll tell you an example or two, of how this is enormously helpful in being adaptive, in learning about which therapies are effective for which patients, and modifying the design as you're going along to adapt to what you learn. Usually there's two arms, and usually the patient is very narrowly defined as triple negative breast cancer in women older than whatever. The sample size is usually fixed in advance, except for a few interim analyses or one or two. Statisticians will slap your hands if you try to look too often. And so clinical research is really weird from a scientific perspective. And what I'm going to tell you about is revolutionary. You actually, you know, look at the data and adapt to what you're seeing. So it's the scientific method. Instead of you close your eyes, open them 5 years from now and say, oh, shucks, we should have done something different. Or, increasingly the case in cancer, nobody cares what the answer is, because we've seen, you know, as I've been speaking, some biologist has targeted another biomarker with a therapy built a monoclonal antibody to hit that target. It's changing incredibly fast. This is my one-picture definition of an adaptive trial. For those of you on an airplane later today, the difference between an adaptive trial and this pilot is that we have protocols. Now, maybe he had a protocol. The protocol said, if the propeller stops, get out and turn it. And my one picture of a platform trial, this is in the sense of drugs, where different drug companies participate in the same process of evaluation through this adaptively randomized thing against other companies, which is a new thing in medical research. For you, these platforms are a lot easier to get organized because they're interventions, and you can build interventions and modify interventions within the context and pair one intervention with another, maybe combinations of interventions. And we try to do that too, but it's really difficult because of IP and using two experimental therapies at the same time, for example. I'm going to tell you a bit about the FDA. The FDA is enormously important in everything I do and everything in medicine because they of course, regulate drugs, for example, and biologics and medical devices. And Janet Woodcock, who's the director of the Center for Drugs at the FDA, very influential person, has been for a long time, is pushing this whole thing that I'm telling you about. And others are as well, others at the FDA, although it's a mixed bag. It's a very conservative group, and they're learning that they can't be as conservative as they once were because biology is passing them by. Several years ago, she said that improved utilization of adaptive and Bayesian methods could help lower the failure rate and expense of phase 3 trials. We are now, phase 3 trials are the trials that one uses for registration, for marketing approval, and they're enormously expensive to run. As a result, as you no doubt have heard, cancer therapies are enormously expensive. I just worked on an Amgen drug that for acute lymphoblastic leukemia that costs $170,000 a year per patient. In 2011, she published a New England Journal of Medicine perspective where she touted this trial that would be looking at individual patients and the response in the individual patients and trying to learn from those patients for future patients and modifying the design accordingly, all about precision medicine. It's called I-SPY 2, and I'm co-PI and designed the trial. And I'm going to tell you about that, but it's also, I-SPY 2 now has clones in all sorts of diseases, many cancers, in Alzheimer's, in Ebola, in community-acquired pneumonia, et cetera. 2013, she said that the FDA will have to turn the clinical trial paradigm on its head to allow personalized drug therapies to get on the market faster. She doesn't know how to do that. I don't know how to do that. Nobody does, but we're experimenting with the experiment, something new. The randomized trial is 75 years old. Hasn't changed much over the course of that time. You name another technology in the modern era that hasn't changed in 75 years. I mean, just take anything out of your pocket and you'll say something has to give, and something is giving. So one of the ways that she thinks things should change is this master protocol in which you look at multiple therapies for a single disease, or single treatment in multiple diseases, or the combination of those, which is I-SPY 2. And this is the future. She wrote an article just last year on master protocols in the New England Journal and illustrates the concept with I-SPY 2. And this is one of the principles that they announced. Two types of innovation are hallmarks of master protocols: the use of a trial network with infrastructure in place to streamline trial logistics, improve data quality, facilitate data collection and sharing, use a common protocol that incorporates innovative statistical approaches to study design and data analysis, enabling a broader set of objectives to be met more effectively than would be possible in independent trials. So it's looking at lots of therapies, and there's adaptive randomization that I'm going to tell you about, but simply comparing many therapies to a single control arm is hugely efficient. I mean, instead of having 10 randomized trials where there's 2-arm trials, a total of 20 arms, 20 times whatever the sample size, versus 11 because you've got 10 experimental arms and 1 control arm that you're using over the course of time, it's hugely efficient. And then we try to do things that are more clever than simple randomization. Scott Gottlieb touted when he was confirmed for the new commissioner of the FDA a trial that I also designed, GBM Agile, where it takes I-SPY 2 one step further and into the registration realm. It's a phase 3 trial, and it has a confirmatory cohort that after you do the adaptive randomization stuff, there's a small cohort of patients that you equally randomize, 50 patients assigned to therapy in order to see that what you've seen before is actually confirmed. GBM is glioblastoma. It's the disease that's killing John McCain and killed Beau Biden, Ted Kennedy, and other non-politicians as well. Companies are seeing the writing on the wall and participating in I-SPY 2, 11 companies, 14 therapies so far. We've graduated 6 therapies, and it's been picked up by the Innovative Medicines Initiative in Europe. Europe is really big on these kind of platform trials, as is Australia and New Zealand, and even China is getting involved. As I said, many cancers and lots of other diseases. And hopefully I'll add home visiting here at some point. So a little bit about I-SPY 2. During the trial, drugs, this is the FDA, drugs are individually targeted to the biology of each woman's tumor. By applying an innovative trial design, researchers then use data from one set of patients' treatments to decide treatment for future women who join the trial. This will help the researchers learn more quickly which investigational drugs will be most beneficial for women with certain biomarkers, precision medicine. There are huge advantages. I knew this thing about comparing to a common control, but I really didn't appreciate the advantages of having many arms in the same trial. And I'll give you a couple of examples of that. This is the New England Journal a few years ago had an article, had a whole issue actually dedicated to I-SPY 2, where the first 2 graduates were announced, and 2 other papers talking about I-SPY 2 and the concept. This is the way there are no p-values in those papers. There are probability distributions, posterior probability distributions of the benefit, and in particular on the benefit of pathologic complete response. I should say a little bit about the trial. It's breast cancer, but it's breast cancer in the modern era, where the old-fashioned way, and still quite common, is you remove the tumor from the breast, deliver systemic therapy, and then wait for some period of time for a long follow-up to see if it comes back. So it's a difficult inferential process because there's nothing happening in between with respect to the cancer until it comes back, which is obviously late and bad. The neoadjuvant approach that we use in I-SPY 2 and pioneered in many ways this approach is leave the tumor in the breast, deliver systemic therapy. After the therapy, remove the tumor, or what was once the tumor, the tissue that was once the tumor, and if it's gone, that's a pathologic complete response. And whether you get a pathologic complete response depends, of course, on the therapy, but it really depends in a crucial way on the biomarker characteristics of that tumor, something that we want to consider. And indeed, we expect, and it's true, that we see one therapy that's good for one biomarker subset, not good for another, but then there are other therapies that are good for those other biomarker subsets. So the idea is to identify, to pair therapies with characteristics of the tumor. So this was, neratinib was one of the drugs that was added to standard therapy, and it had a pathologic complete response rate in its graduating signature of 56%. 56% of the women who were assigned in this tumor type benefited from neratinib in comparison to the control, which was only 33%. And the reason I'm telling you this is because during the course of the trial, we're looking at the data continually. There's an algorithm that says this is the new randomization probabilities for neratinib and the other arms that are in the trial. And in one subset, it said 0 probability. It said this drug is not working in this subset. On the other hand, in another subset, it graduated. It did, what I just indicated to you. And the DSMB said to me, DSMB meets monthly, which is unusual for a data monitoring committee. And they said to me at one monthly visit, the drug is not doing very well in this subset of women. Maybe we should drop it. And I said, that's a good idea, but the algorithm has already beaten you to the punch. It is assigning with 0 probability. These are the signatures we consider. These are 10 possible signatures. This one, for example, is triple negative disease, which is, you may, if you've read anything about breast cancer, you've read about this. There are 10 such signatures. This is the one that had 0. This is the group that had 0 probability. These were the groups that were getting high probability, and eventually the drug graduated in that subset. Those two combinations, HER2 positive, hormone receptor negative. If this were a standard trial, this would be a nightmare. You'd have to amend the trial. You'd have to announce that the drug is not doing very well in these, and you're not including these women anymore. We didn't have to do that. In fact, nobody knew that these zeros were there until afterwards, because we had other arms to assign to those women. So the benefit of having these many arms. Another example of the benefit, and I'll not show you pictures of this, but during the course of the trial, which we started in 2010, a drug called pertuzumab was approved. It received accelerated approval from the FDA in a particular subset of women. It happened that we had that drug in the trial, but we were comparing to a control arm that didn't have that drug. Our patients, our physicians, our data monitoring committee said, you have to drop the standard arm. What? If we drop the standard, what would that do to a 2-arm trial? It's over. We just said, all right, let's use the controls that we've had. We've got this lamination of the arms. And so even though we're assigning patients now to pertuzumab instead of that standard arm, we've got pertuzumab that's being compared to the experimental arm here. We've got other arms in the trial that were compared back against the control arm when they were concurrently being randomized. So even though A is not randomized against B, A is randomized against C, which is randomized previously against B, which is randomized against D, and E is, you get the picture. We build the model, we're able to use all of the controls in the trial. And that, by the way, with discounting, and that, by the way, characterizes our phase 3 trials, which is, nobody has ever heard of trying to do this in a phase 3 trial, but the FDA has agreed. So this is the adaptive process, just to tell you about some of the drugs that have graduated. 6 of them have graduated in different biomarker subsets, some overlap, but with different sample sizes. We stop when we get there because our eyes are open rather than closed and going to a particular sample size. One more example, pembrolizumab, which you no doubt have seen football games with, this is Keytruda or Opdivo. These are 2 almost identical drugs. The big deal in cancer these days, immunotherapy, and hugely effective. Pembrolizumab, Keytruda has 800 trials going on as we speak, evaluating things. I-SPY 2 was one of them. It was in the trial in HER2 negative disease, and it showed amazing benefit. This in triple negative disease, it showed a 60% a pathologic complete response rate as opposed to control of 20%. And the interesting thing, the reason I tell you this, is because it graduated when there was 1 patient through surgery. 1 patient had the primary endpoint. And I looked at the algorithm and it was telling me pembrolizumab graduates. You should stop accruing to it and let the folks at Merck know, and 6 months from now announce the results, et cetera. And I said to the DMC, you know, I can't go to Merck and tell them that the drug graduated with 1 outcome. It was a PCR, it was a path CR, by the way. What did this? What did it is in I-SPY 2 and in these other trials, we have a longitudinal model, and it's looking at MRIs over the course of time in the individual women, and it is predicting whether that woman is going to achieve a path CR. The MRI is far from perfect. But when I went in, I of course went in and looked at what the data were, and the MRIs were fantastic. There were like 12 patients who had had 12 weeks, and another an MRI at 12 weeks, and another dozen or so patients who had MRIs at 3 weeks, and they were telling me that the tumor was melting, more than a 50% decrease in just 3 weeks. And so it was predicting that these were going to be path CRs. So I said, let's accrue for another 1 minute, accrue for another period of time, and then go back and then see, you know, we'll have more data in, and the next month there were another 12 patients with data in, and 11 of them were path CRs. So we stopped accrual, and what it was predicting at this time with 1 patient in was a path CR rate of 60% for pembrolizumab and 20% for control. And when we announced, after a total of 69 patients on the drug, it was 29 in this particular subset, it was 60% on pembrolizumab and 20% on control, but with much tighter densities. So what, if anything, should I be telling you more? This is, you can get my slides and see what I do with the GBM Agile in pancreatic cancer. 2 other, but these are phase 3 trials that have this cohort that I told you about. And I'm going to go to the very last slide to say you have to do simulations. If you build a trial like this, you have to do modeling and simulations so that you understand what type 1 error is, what the power is, but more importantly, to communicate to people, IRBs, regulators, what is this trial about? So what I do is I show them not 10,000 simulations, which I need for power calculations, but so they can see how the trial works. I show them 1 simulation, a second maybe 5 simulations, and they get an idea. Okay, so this is what the trial is doing. What I hope for and get is data monitoring committees that say to me, you know we don't control the design of the trial, but if we could control the design of the trial, we'd do exactly what the trial is doing. And when I show simulations, people see, this is what it's doing. Do I like that? If these were the data, would I want to do that? And if not, we'll go back and make the investigators and the regulators happy. So I'll stop there. Thank you.